Hello everyone, let's talk about Parazoa and Eumetazoa today. In this and next 11 lectures, I will present the great diversity of animals. I start with the morphologically simplest members of the animal kingdom: sponges, jellyfish, and some types of worms. Despite their simplicity, these animals can carry out all the essential functions of life, just as do more morphologically complex animals, eating, respiring, reproducing, and protecting themselves. The major organization of the animal body first evolved in these animals. The basic body plan from which all the rest of animals evolved. Systematists traditionally divided the metazoa into two main branches. Parazoa comprises animals that do not possess tissues. This is the phylum Porifera. Because they are so different in so many ways from other animals, some scientists inferred that sponges were not closely related to other animals, which would mean that what we consider animals had two separate origins. Eumetazoa are animals that have a definite shape and symmetry. All have tissues, and most have organs and organ systems. Most systematists agree that Parazoa and Eumetazoa are descended from a common ancestor, so animal life had a single origin. Most phylogenies constructed with molecular data consider Parazoa to be at the base of the animal tree of life. The Eumetazoa contains animals that evolved the first key transition in the animal body plan: distinct tissues. The embryonic cell layers differentiate into the tissues of the adult body, giving rise to the body plan characteristic of each group of animals. In the Bilateria, the embryonic mesoderm forms the muscles. Eumetazoans also evolve body symmetry, the two main types of which are radial and bilateral symmetry. The Porifera, or sponges, are the simplest of the many-celled animals. They lack defined tissues and are thus excluded from metazoa. They are mainly marine, sessile filter feeders. They may be encrusting and irregular in shape, but many have a definite form, for example, bowl or vase-shaped with approximate radial symmetry. But most members of this phylum lack symmetry. The skeleton is internal and may be organic, or made of opaline silica, or calcite, or aragonite. Only those with a mineral skeleton are likely to become fossils. A sponge has no true tissues, being essentially an aggregation of a few types of cells. The simplest form is sac-shaped. The body wall consists of an inner and outer cell layer sandwiching a jelly-like substance containing wandering cells. The latter secretes the spicules of which the skeleton is composed. The spicules may remain discrete and thus separate on the death of the animal, or they may unite to form a rigid structure which may be preserved. The body wall is perforated on the outside by small pores leading by a system of canals to the central cavity. This is lined by special feeding collar cells that have thread-like flagella. These beat to and fro, creating a current of water, which is drawn in through the pores into the central cavity and passed out by one or more large openings on the upper surface. In the process, oxygen is absorbed, food particles are filtered out by the collar cells, and waste is carried away. Each collar cell resembles a protist with a single flagellum, a similarity that may reflect its evolutionary derivation. The pressure created by the beating flagella in the cavity contributes to circulating the water that brings in food and oxygen and carries out wastes. In large sponges, the inner wall of the body interior is convoluted, increasing the surface area and therefore the number of flagella. In such a sponge, 1 cubic centimeter of sponge can propel more than 20 liters of water per day. Collar cells also capture food particles from the passing water, engulfing and digesting them. Obviously, this arrangement restricts a sponge to feeding on particles considerably smaller than collar cells, largely bacteria. Siphonia. The skeleton is tulip-shaped and was attached by a stalk. Minute surface pores lead to a system of canals which transfers the thick wall. Some is radial direction and others parallel to the outer surface. Spicules tightly packed or irregular shape, siliceous. Ventriculitis. The skeleton is roughly vase to bowl shaped, varying from deep to shallow, and was attached by rootlets. The pores lead to simple radial canals. Spicules 6-rayed, crossing at right angles, siliceous. Raphidonemma. The skeleton is an open vase or funnel shape, thick-walled with rough outer surface. Canal system rather indistinct. Spicules 1 and 3-rayed, calcareous. Sclerosponges, coralline sponges recently discovered living deep down in the coral reefs of Jamaica, have a skeleton or organic fibers, needle-like siliceous spicules, and aragonite. The latter is secreted as a compact base into the top of which the soft body penetrates via minute pits. Traces of these pits are preserved as narrow tubes with horizontal partitions within the base, and silica spicules may also be entrapped. The living tissue is a thin veneer over the base and has a knobbly surface, with many small incurrent pores lying in depressions between domes where larger excurrent pores open. Water drawn into the sponge is channeled along radially arranged canal systems, each converging to open in an excurrent pore. Sponges are gregarious, living for the most part attached to a hard substrate in relatively clear water. Calcareous sponges live in reasonably shuttered, warm, shallow waters, less than 100 meters deep. Fossil forms show similar preferences. The siliceous sponges range in deep water, most occurring between 100 and 400 meters. Those with 6-rayed spicules, the glass sponges, are also found in deep water, down to about 6,000 meters, living on a muddy substrate. Generally, sponges are abundant only locally and occur mainly in shallow water rocks such as sandstones and limestones, including reefs. Paleozoic sponges were predominantly siliceous forms and are commonly represented by isolated spicules in charts and silicified limestones. The Cnidaria include the simplest of the metazoa in which definite tissues are developed. Examples include the jellyfish and sea anemones, which are soft-bodied, and the reef-building corals, which secrete an external calcareous skeleton. There are also a variety of extinct corals. Cnidarians are exclusively aquatic, and the majority are marine. They may be sessile or free-swimming. The sessile form, or polyp, may occur as a single individual or be united with others to form a colony. Basically, it is sac-shaped and is attached at its base to the seafloor with its one opening, the mouth, at its upper free end. The free-swimming form, or medusa, resembles an umbrella with tentacles hanging down around the margin. Some cnidarians, like corals, exist only as polyps, while in others the life history involves an alternation of polyp and medusoid generations, the one giving rise to the other. The cnidarian body is of simple structure consisting of an outer layer, ectoderm, and an inner layer, endoderm, which in more advanced members, like the corals, are separated by a jelly-like supportive substance, mesoglia. The body wall encloses a central cavity, the gut or enteron, and this may be divided by radial partitions, the mesenteries, which aid in digestion and absorption of food. The mouth serves for both intake of food and discharge of waste and larvae. It is surrounded by retractile tentacles, which are armed with nematocysts, microscopic intracellular structures unique to the phylum, with which cnidarians capture their prey. There is no blood system, and the nervous system is a diffuse network of cells. Reproduction is sexual or asexual. Typically, the body shows radial symmetry, the parts of the body being repeated around the mouth. Cnidarians are common in warm, shallow seas, but some forms live at depths of down to 6,000 meters, and in temperatures as low as 1 degree. Although often referred to as stinging cells, nematocysts are capsules, each secreted within a cell called a nematocyte. Cnidarians may be morphologically simple, but nematocysts are the most complex structures secreted by single animal cells. When appropriately stimulated, the closure at the end of the capsule springs open, and the tubule is emitted. Nematocyte discharge is one of the fastest cellular processes in nature. Although the action of nematocytes is often described as a harpoon-like, the tubule everts, it may penetrate or wrap around an object. The tubuli of some nematocysts are barbed and some carry venom. Only the anthozoan corals are common as fossils, and only the rugose corals are considered in detail here. Anthozoans exist only as polyps, either solitary or as colonies. The enteron is divided by 6, 8, or more mesenteries, which may be paired. They are all marine. The rugose corals are solitary or compound Paleozoic corals with a calcareous skeleton, the corallum, divided by vertical radial partitions, the septa. Zephranthitis. solitary, horn-shaped with deep callus and the cardinal fossa on the concave side. Cardinal fossa, a distinct elongated space bonded laterally by major septa, and the short cardinal septum projects into it. Usually no minor septa and no disciplements. Tabulae rather irregular and inversely conical in shape. The rugosa appeared in the mid-Ordovician times, and by the Silurian were common and widespread. They were, however, subordinate to the tabulate corals and stromatoporoids in the clear water calcareous phases. In the later Devonian numbers plummeted, and many families became extinct. There was, however, a dramatic recovery in the lower Carboniferous with the arrival of new families, some showing new features like complex axial structures not seen in earlier forms. They persisted until the end of Permian. The tabulata are extinct compound corals in which the corallum is built up of slender corallites partitioned transversely by many tabulae. Tabulate corals appear in the early Ordovician and became varied and widespread in the latter part of that period, finally disappearing at the close of the Permian period. They occur most common in shallow water limestones and calcareous shales, and played a significant role in reef formation in the Silurian and Devonian. They outnumbered rugose corals in reefs of that time. The Scleractinia are solitary or colonial corals with an aragonite skeleton in which septa form cycles of 6 or multiples of 6. They first appeared in the mid-Triassic and are important reef formers in tropical seas today. Sponges possess multicellularity but have neither tissue-level development nor body symmetry. Cells that compose a sponge include a layer of collar cells, a layer of epithelial cells, and amoeboid cells in the mesohyl between the two layers. Collar cells have flagella that beat to circulate water through the sponge body, allowing food particles to be trapped. Members of phylum Cnidaria have nematocysts, capsules used in defense and prey capture, and are radially symmetrical. That is all for today. Thanks for watching. See you next time.